Dear learners, welcome back to NPTEL online certification course on novel drug delivery systems. I am Dr. Satish Devanapalli, Assistant Professor at Department of Pharmaceutical Science and Technology, Institute of Chemical Technology, Mumbai. So, in our novel drug delivery systems, we are learning the topic on protein, peptide and nucleic acid delivery systems. In our previous class, we have discussed the topic, that is introduction to protein peptide delivery systems. In that, we learn the basics of protein peptide molecules, and then we have studied the difference between proteins and the peptides, and also we have seen the different structures of the proteins, and then we have also studied the functions of proteins and peptides. In addition to that, we learn the therapeutic uses of the protein peptide drugs, and after that we have seen the challenges which comes during the delivery of the proteins and peptide formulations. Not only that, we have seen what are the instability issues with the proteins and peptides. So, today we are going to see some new topics on the protein peptide drug delivery systems. So, here we will start with the key considerations for protein peptide delivery systems. There we are going to understand what are the physicochemical properties of the protein molecules and peptide molecules which are considered as in a delivery systems. And after that, we will see what are the various barriers which will hinder the delivery of the protein and peptide molecules. And if time permits, we will see the third topic and we will go further on the techniques for stabilization of the protein peptide molecules. Then we will see the important components used in the protein peptide formulations, and further we will see protein peptide delivery systems, and 3 dedicated lectures I will focus on these 3 topics. So, first we will start with the key consideration for protein peptide delivery systems, and try to understand in our previous lectures also. If you see any type of formulation aspects, we need to understand the nature of the molecule. So, if you can see as an example, in case of gastroretentive drug delivery systems, even in the transdermal drug delivery system and other type of mucoadhesive systems, in all the cases we understood the nature of the molecule, because whenever you are targeting to any site or if you want to make any formulation, we know that we are the pharmacy candidates and we should do the pre-formulation studies. And again we know that pre-formulation means nothing but understanding the physicochemical properties of the drugs. When it comes to the proteins and peptide, yes, whenever you want to make any formulation, you must understand the nature of the molecule. So, here also similar to the drugs and other molecules, it may be biological molecules. So, here also we need to understand the physicochemical properties of the proteins. So, yes, we should know the molecular weight of the proteins and peptides, and also ionization constant and hydrophobicity, the pH and stability, enzyme degradation also. So, these are the few important physicochemical properties we need to understand. Based on that, we can formulate a novel delivery system. Consider we can go back to the like pharmaceutical formulations or which are API or drug related formulations. Yes, we do lot of physicochemical properties, right. So, for example, in case of physical properties, in the pre-formulation studies, we will try to understand what is the solubility of the molecule, and also we see what is the pKa and the log P, right, and we see some of the flow properties. So, these are all related to the physical properties of the drug molecules, and when it comes to the chemical properties, so we try to understand the nature of the molecule. It may be in terms of like we can say like crystallinity of the molecule, chirality, right. So, other type of the chemical properties, amorphous form, right. So, these all are very important, which will directly indirectly influence the fate of the formulation. So, once you know these physical and chemical properties, it will be very easy to create the formulation. We can also do some of the stability parameters. There we can understand the stability of the molecule by doing of oxidation studies and the hydrolysis and photolysis. So, with this we can understand the nature of the molecule. If any problem arise, for example, consider you are facing a solubility issue. So, here what we do, we try to improve the solubility of the drug molecules. Similarly, here also for any biological molecule, you should know before developing any formulation, you should know the molecular weight, ionization constant, hydrophobicity, pH, stability and enzymatical degradation. We will see one by one how which will influence the delivery. So, the first point comes here, it is the molecular weight. For a key consideration, yes, the how it will impact a peptide's size affects the ability to cross biological membrane, reach its target. Proteins with their larger molecular weights have greater difficulty in oral absorption compared to small peptides. Yes, as we know that when you are going for oral absorption, and yes, that is one of the major limitation in the biological molecules which are comes with the high molecular weight. Also we learned the same topic in our previous session, right. If any molecule which is greater than 500 Daltons, it is very difficult to permeate through the membranes. So, that is a molecular weight has an impact on the permeability as well as absorption. So, how to do that? A smaller molecular weight peptides are generally easier to deliver, particularly orally especially, but may lack the structural complexity of the larger proteins. So, whenever you have small molecular weight, the absorption is very easy because they were able to cross the biological membranes, whereas it comes in the large molecules. In such cases, larger proteins require specialized delivery methods, such as you can convert your oral formulations into the injections, either intravenous or intramuscular, some cases subcutaneous also possible. And you can also convert your molecules in the carriers by using of nanocarriers, either it may be polymeric or it may be lipidic formulations. There you can convert, or also you can use some of the emulsions which will also help for as a carrier. And yes, you can also use cell penetrating peptide, which is nothing but this will help as a carrier. We need to conjugate your protein with this peptide. Okay. So, this peptide is nothing but CPP, nothing but cell penetrating peptide. So, this will help for the penetration of the molecule. So, that will give the entry into the cells. Whenever you have a large molecules, you need to go through either injection or by using carriers or by complexation with cell penetrating peptide that will take the molecule into the deeper layers of the cells. Okay. So, molecular weight has a major consideration in the delivery of the proteins and peptides. Now, second is we know that ionization constant, similarly this is required for even for the drugs. Yes, for an absorption your molecule should show 50% in ionized form and anionized form, right. Here the pH has a major role. We need to try to identify the pH where you are getting the dissociation. So, that is called as dissociation constant, or you can say ionization constant. It is also known as pKa value. We know very well in the pre-formulation of the pharmaceutical drugs and other type of new chemical NCEs. So, here we can see the what is the impact of ionization constant in protein drug delivery. A peptide's pKa determines its charge at a given pH. Of course, at that particular pH your molecule will dissociate into ionized form as well as unionized form. So, that is very very important for the absorption as well as solubility, which affects solubility and interaction with the charged surfaces, ability to penetrate biological barriers. So, that is why we need to know the ionization constant of your molecule before developing formulation. Now, as we discussed in these slides, you can see the consideration solubility, the charge state of peptide significantly impacts its solubility in aqueous environment. So, for that dissociation constant is very very important, and also membrane permeability. Charged peptides may have more difficult passing through bilayers, yes, because they may interact with the membranes, and yes, some cases like a positive charge which can interact with the negative charge of the lipid membranes, which will help for the high retention, that may also allow for the long time effect. And third one is target binding. The ionization of peptides functional group is crucial for its binding affinity, receptors as well as other biological targets. If it is a ionization is depended on the particular pH, and if that functional groups if may help, like for example, you are taking an NH2 groups, okay, and in the low pH it will show the positive charge, and that will also help to interact with the negative charge of the membranes. So, it always advantages for the target binding. So, ionization constant is very very important for the solubility as well as permeability and the target binding. And the third one is hydrophobicity. The third consideration for protein delivery is the hydrophobicity. Yes, this is very very important because that will balance the hydrophobic as well as hydrophilic properties of the amino acid, which will definitely influence the aggregation, membrane interaction, its ability to interact with the hydrophobic cellular environments. And the consideration point is that, so peptides often have exposed hydrophobic groups than the proteins, because their percentage of amino acids is very less and the exposure of the hydrophobic groups will be very very high compared with the proteins, and that hydrophobicity percentage will also influence the solubility if hydrophobicity is very high, then definitely you will see the solubility issues of the molecules. And not only that, that surface properties also very important, that will also depends on the hydrophobicity of molecule, and this hydrophobicity also will influence the permeability of molecule. Yes, hydrophobicity or you can say lipophilicity is very important for permeability, but extreme hydrophobicity is not suitable, that will also affect the solubility as well as some stability issues. And this hydrophobicity also important to maintain the stability and for the high efficacy within the formulation. So, the third consideration, hydrophobicity, is very important for a protein peptide molecule, and before designing of any formulation we should understand the nature of the protein and peptides. Third one is pH related consideration. Now, here definitely as we are learning, it has a good impact in terms of solubility as well as pKa and stability of the formulation. So, here we are going to see three different points. The first one is pH related consideration and solubility. As we know that the solubility is directly dependent on the pH, and there are the molecules which even in the pharmaceutical platform, there are the drugs. The drugs can be classified into two categories, weak acids and weak bases, and definitely these two type of category of the molecules, the solubility is dependent on the pH, right. So, in here also in the protein peptides, so solubility also depend on the pH. So, peptide solubility is highly pH dependent, typically exhibiting minimum solubility at isoelectronic point. So, isoelectric point peptide solubility is highly pH dependent, typically exhibiting minimum solubility at the isoelectric point. So, isoelectric point is nothing but it is the pH where the amino acids or proteins or peptides will show the net charge is 0. So, we can understand by plotting of simple this graph. In x axis you can take pH and you can here take the charge, and now you can consider this a plus and here is the negative charge. Okay. So, isoelectric point is like this. So, this is the isoelectric point, consider this a pH 5.5. So, here you will see the below isoelectric point the amino acids will show the positive charge, and below the above the isoelectric point it will show the negative charge. So, this is the point, the pH, that is called as isoelectric point. There the net charge of the amino acid is 0, that is called as isoelectric point. There it will, the solubility will be very less. So, we need to identify the isoelectric point of the biological molecule, right. So, there are the amino acids and the proteins, their isoelectric point the near to 5.5 and 6. There are the amino acids and the protein peptides comes isoelectric point, it will go into the basic pH also. So, you need to understand, otherwise you will see lot of solubility problems while developing the formulation. If you know that isoelectric point, based on that you can select the right pH medium for the solubility and further formulation studies. And the second is pH is related to considered for developing of the formulation. pH modifiers, buffers are crucial for stabilizing the peptide formulations. We will see in our upcoming slides. So, there we are going to see what is the role of pH as well as buffers, which will directly impact on the stability of the formulation. So, but careful selection is needed as some acidifiers may cause peptide degradation, precipitation of other formulation components. So, we need to study the compatibility of the proteins and peptides with the pH modifiers as well as buffers. Based on the observation, we need to select right type of pH modifier and buffers, so that we can avoid the peptide degradation as well as precipitation, any other type of instability issues with the selection. So, the third point is in vivo environment. Yes, in the body also we have different pH conditions which varies from the oral, from the mouth to stomach and small intestine and large intestine. And we have variety of the pHs, like in the blood we have pH 7.4 and ocular we prefer 7.4, oral we prefer 6.8, when it comes to the skin we prefer 7.4, around 6.8 also. So, we have so much variation within the body, right. So, in vivo environment nothing but the pH of the targeting site also very important for your molecule. So, you need to understand the physiological environment as well as the pH dependent study or solubility and formulation also is very important. Now, you can see for oral delivery, low pH of the gastric fluid can be concerned. Why? Because that will induce the degradation, like acidic hydrolysis also can happens. So, that we should know the stability issues of your molecule. So, the acidic it may comes around 1.5 to 2 is the pH, right. So, for that case we need to come up with the various types of the formulation to overcome the oral delivery. And while higher pH in the small intestine can cause issue of peptides formulated in certain coatings. Yes. So, what you can do when you want to go for the to overcome the acidic degradation, you can use enteric coated polymers or enteric coating process, so that you can avoid the harsh acidic condition degradation of the molecule by making into the enteric coated. So, there they will sensitize and they will undergo stimulation at particular pH which is higher than acidic condition, then your drug may release out from the carrier and then it will show its activity. And those susceptible to alkaline conditions, nothing but that coatings may undergo the degradation and then your drug will release out. So, based on that you can modify your formulations. And fifth one, we are going to see stability related consideration. This already we discussed about the what are the physical instability issues which already discussed, physical instability issues, chemical instability issues and the external factors. So, here you can see the aggregation and the denaturation which will directly indirectly affect the activity of the therapeutic molecules. It will completely lose its activity. So, deamidation is one of the problem and also we have seen oxidation in our previous lecture, and also external factors, temperature, agitation, shear during the formulation. So, while using of any other chemicals, even in the process, even in the process factors will definitely will degrade your molecule. So, which are considered as a instability conditions. So, yes, for that whenever you are facing any problem related to the stability, you can use some of the considerations which are you can use either stabilizing agents. So, which are nothing but we can use buffers and the enzyme degradation inhibitors which are nothing but peptidase inhibitors, and other excipients to control the pH and protect against the degradation. So, here you can use pH modifiers also. There are the pH modifiers. So, they will modify the pH of the environment. Why we need to use pH modification? There are the peptide degradation enzymes. They are susceptible, they will survive in particular pH, consider which they are surviving in low pH. So, what we can do by using of pH modifiers, we can increase the pH from low to near to 7.4 or around 5.5. So, enzymes that protein peptide degrading enzymes, we can inhibit by modifying the pH of the environment. That is the first step. And then the second thing is formulation approach. So, here we can use technique like lipid based formulations, micro and nanoparticles, and also there are the formulation like hydrogels. We can also protect the molecules from the degradation. So, by using of all these techniques, okay, we can avoid the degradation. So, yes, sixth point we can discuss here, enzymatic degradation related consideration. So, this may happen even from oral, even from the if you go to pulmonary, also which may happens in the parenteral also. So, these enzymes, plasma enzymes and the gastric enzymes will degrade the molecule. Definitely you have to use enzyme degradation inhibitors. Okay. So, first consideration pH modification, as I mentioned, we can inactivate the stomach enzymes like pepsin. Also we can incorporate enzymatic inhibitors, so which will block the enzymes, and the protective formulations like enteric coating, there we can use cellulose acetate phthalate. Also you can use hydroxypropyl methyl cellulose phthalate. So, such phthalate derivatives can be used for enteric coating. Nanoparticles, definitely that will protect from all type of degradation, it will also help for the control release, and we can also use for the targeting. Then pegylation using of PEG molecule, that is nothing but polyethylene glycol, we can conjugate with the protein and peptides. So, that will help and that will prevent the degradation of the enzyme because of this linkage. So, that will help conjugation to molecules like albumin also possible. So, here we can use albumin, which are called as the polymer protein, or you can say peptide conjugates. So, this will the ADME profile of this molecule is completely different from the native molecule. So, with that we can improve the stability as well as circulation time. So, with this we are going to stop the lecture here. So far we discussed what are the key considerations on protein drug delivery systems. In our upcoming classes we will see the remaining topics on the barriers and the key components used and also formulation delivery approaches in the protein drug delivery systems. Thank you so much. See you soon.